speaker, Dr. Tom Reh. Dr. Reh received his bachelor degree in biochemistry from the University of Illinois Urbana-Champaign and his PhD in neuroscience from the University of Wisconsin-Madison. Then he went on to postdoctoral studies at Princeton and became an assistant professor at University of Calgary. And he is now currently a professor of biological structure at the University of Washington. Dr. Reh and his lab have been making important contributions in the field of retinal regeneration and development. He has been working at the interface between the development and regeneration of the eye with the goal of applying the principles from the developmental biology to develop strategies for promoting retinal regeneration. His research has been widely recognized by a number of publications, including Nature and Nature Neuroscience, and he has received many fundings from NIH and other private foundations, and he has served on several national and international grant review panels and is currently a member of Scientific Advisory Board of the Foundation Fighting Blindness. And Dr. Reh has received several awards for his work, including Alberta Heritage Foundation for Medical Research and Sloan Scholar Awards. Today, we are very excited to hear more about his research. So everyone, please welcome Dr. Reh.
Okay, and I will share my screen and away we go. I think that. Hey everybody, so thanks for the invite, Chris. I realized that, yeah, we've had a lot of great influences over our life, and scientifically, when Chris was here at the University of Washington, I wouldn't say we had, he was a scientific mentor, but he definitely always was entertaining to listen to. Nevertheless, we miss him since he left. It's been many years, but it was great when you were here, Chris. Still, I see that your career's only gotten better since you left, so that may say something about the university. I would like to start by also just giving a little backstory of the photo here, just because in this era of pandemics and all, this photo is hopefully the sign of changes to come, and maybe we'll all be able to get together in real time from now on, because what I actually sent was this photo where I got to hang out with my granddaughter a couple weeks ago for the first time in a year at the Canadian Peace Arch, because she lives in Canada and the only way I can get to see her is if we just hang out at the border. But nevertheless, it was great fun, and like I say, I hope this is a harbinger of changes to come, and we'll be able to get together in person again pretty soon. What I'm going to talk about today is our work on stimulating regeneration in the retina. And this has been, I guess you could say, something I've been fascinated with, or maybe even obsessed by most of my career. The retina, like the rest of the central nervous system, we know it doesn't regenerate its neurons in mammals very well at all, if at all. And there's only a couple places in the entire central nervous system that have ongoing neurogenesis into adulthood. Nevertheless, or maybe as a consequence, I guess you could say, many diseases that affect the retina lead to loss of one or more of the different retinal cell types. And I don't really need to talk to this crowd about all the different diseases and what cell types they affect, but I'm going to come back to this at the end of the talk. But just suffice it to say that once these cells are lost, we don't naturally replace them in our eyes, and so patients have visual impairment if any one or more of these cell types degenerate from one of these diseases. Now, this is actually a slide I didn't know Sanai was going to be listening today, but it's something that Sanai did years ago when she was in the lab. But it just highlights to me these the idea that a retina can regenerate is somewhat, to a clinician might seem completely, impossible, but to an amphibian, it's not a problem at all. The retina regenerates completely and functions again after it's pulled out of his eye. So essentially what you do is you just make a little slit across the cornea and then pull out the retina. And then within a few weeks, it starts to, there's a lot of proliferation that goes on from the pigmented epithelium. And then, sorry about that. And then the pigment cells go on to depigment and become retinal progenitors and restore the retina entirely again. And this was known to occur in amphibians for nearly 100 years, but it wasn't thought to occur in birds and mammals. And in fact, as far as we can tell, it does. That process of dedifferentiation of the pigment cells to make a retinal progenitor does not occur in warm-blooded vertebrates. But what's interesting is that when Andy Fisher was in my lab, he discovered that birds, in fact, do show a regenerative response. And the cells of origin are the Müller glia. And here you can see a couple examples of some retinal neuron markers like QCD or calretinin. And what Andy found was that after injecting the NMDA into the chick retina, he could see EDU or BRDU in those days being incorporated into the dividing Müller glia, and then some of their progeny would differentiate these neuronal cell types. And this was something we did years ago, and it was really the first evidence that Müller glia could provide a source for new neurons. And this was basically the experimental paradigm, making an injection of NMDA and following up with BRDU. And you could see a robust regenerative response or robust proliferative response, followed by a subset of these new cells differentiating into neurons. And again, this is just repeating that, but we were able to track over time and follow-up work of the onset of new of genes getting expressed in these dedifferentiating Müller glial cells. And we could see this group of genes are typically found in development, including this one, CASH1, which is now called ASCL1. But we could see that that gene, which is normally defines developing progenitors, was turned on after injury. Now, after Andy's studies were published, and he and I followed up on a number of ways, Dan Goldman actually investigated whether Müller glia were the source of regeneration in zebrafish, and he found that indeed they were. And Pam Raymond at that time and several others also had identified that Müller glia re-entered the cell cycle and were able to give rise to new neurons. And here we can see Ryan Thummel, a paper by Ryan Thummel and David Hyde's group, where they destroyed all the rods, and you could see the rods are replaced over the next few weeks very rapidly in the fish. And this comes as a result of cell divisions within the Müller glia, very similar to what happens in the bird retina, in the chick. Now, so this just summarizes what I've said so far, is that the amphibian regenerates from the dedifferentiation of the pigmented epithelium, which is a non-neuronal cell. The fish and the bird regenerate from a Müller glial cell through multiple rounds of division in the fish and through one round of division in the bird. There's a difference between these two animals, though. The fish regenerates all the different types of neurons, and the bird primarily regenerates amacrine cells, though some bipolar cells were found, and a few ganglion cells we could also find at that time. But in the mammal, repeated attempts to try and demonstrate any endogenous capacity for regeneration have really shown very limited, if any, neurogenesis can occur. Nevertheless, we know that Müller glia and retinal neurons share a common lineage. So during development, there's a progenitor cell in the retina that undergoes rounds of division and will generate clones that contain both, say, bipolar and amacrine cells and rod photoreceptors and Müller glia, like what's shown here. This was a lineage trace that Joe Brzezinski did while he was in the lab. And you can see that a clones of cells contain both neurons and glia. So what's the difference between this cell here, which is a progenitor and can generate neurons and glia, and this cell here, which after injury in mammal cannot generate neurons normally, but in fish, it can. And so we explored that actually using an approach years ago by FACS purifying progenitor cells during development in developing retina, and then FACS purifying Müller glia in mature retina, and then taking a look, and we used the same, we used a Hes5 GFP line of mice in order to FACS purify these cells. And then we did a transcriptomic approach, and we found genes that were highly expressed in the newborn mouse progenitors, but were not expressed in the glia. And we thought, okay, if we take these transcription factors, which we're looking at here, and we put them into the mature glia, will they convert the mature glia back into progenitor cells? And this was kind of reprogramming approach was being taken a lot back then. It was really stimulated by Yamanaka's demonstration that you could reprogram fibroblasts into iPSCs, but people were trying attempts at reprogramming many different cells into other cells. And so we basically took a similar reprogramming approach. And so our aim was to express all these transcription factors in glia. We did like this one gene, ASCL1, because Dan Goldman's group had shown that ASCL1 was important in fish regeneration. And by a morpholino to ASCL1, if they injected that into the fish eye around the time, right after the damage, they could really cut into the proliferation and knock it down, and the regeneration was pretty stalled, whereas control morpholinos didn't have that effect. And as I've told you in the chick, the ASCL1 does get expressed after injury. So we thought maybe overexpressing ASCL1 or any of those transcription factors, we sort of did a mini screen, and we asked whether that would stimulate regeneration. Now, we did this first in dissociated cultures of retina. So we went back and we took those Hes5 GFP glia, and we made dissociated cultures of them. And really, Julia Pollack worked out how to culture Müller glia, because it was known from the work of several other earlier investigators that rat Müller glia could be cultured. Particularly Vijay Sarthi had shown that years ago, but nobody had really worked out the culture for mouse Müller glia. And so we tested a variety of different conditions and figured out that we could culture these cells from 2-week-old mice. They would grow pretty well. You could culture them from adult mice, but they work better from 2-week-old mice. So when Julia overexpressed these different transcription factors in the glia, we just initially looked for morphological changes. And I think you can see on the right here, you can see that these cells that got infected with the ASCL1 lentivirus now really start to change in their morphology from the flat glial morphology that you see over on the left to this more neuronal morphology, these small neurons that popped up in the culture. And she was able to do lineage tracing using an RLBP-Cree strategy so that she could show that the new neurons that arose in these cultures actually were derived from the Müller glia. So the RLBP-Cree is only expressed in Müller glia and pigment epithelial cells. And so we dissect away the pigment layer, and so we just grow the retina layer, and then we can label the Müller glia with the RLBP-Cree. We can also use another glial-specific Cree called the GLAST Cree, and we've used both of those Crees and gotten similar results with either one. Now, encouraged by Julia's results in vitro, which we still do to screen factors, and we've, like I said, we've screened many different factors, but ASCL1 was really the most powerful one for reprogramming that we found early in the game. So we developed a tetracycline activator system to activate ASCL1 in vivo, and Yumi Ueki really spearheaded this approach, and we made a transgenic mouse that would do this, and also it turned out that Nakafuku's group had also made a similar mouse. So we've used both mice, one is an RFP and one is a GFP, and worked, and we traded mice and sent him ours. And I think that what you can see though here is the GFP mouse line. You can see all of these cells in the retina, this low power view. These are all Müller glia, and they're expressing the transgene ASCL1. And when we found that if we expressed the transgene ASCL1, it didn't dramatically change the glia in terms of their morphology, and they looked pretty normal. But what Yumi found is if she made an injury to the retina using NMDA, then these glial-looking cells would transform into cells that took on more of a neuronal-like morphology. And so here's a cell that looks quite a bit like an amacrine cell, and this cell here is bipolar-like cell, and then we even saw some cells that have morphology similar to photoreceptors. But the thing she found was that this only worked in mice that were 2 weeks old. If she did the same experiment, starting with mice that were a month or two old, we got no neuronal-like morphologies in the cells. And I should say we could also stain these cells with neuron-specific markers, and we would see, here's an example of some nice glial-derived neurons that have really interesting arborizations. And but then there are also clearly some cells that have completely abnormal morphology. We would have no idea what to classify that cell as by morphology. But you can see that these cells do express markers like QCD, just similar to what the bird did, or CABP5, which is shown in red here. So we can see that they express markers of bipolar cells and amacrine cells. Now, because this only worked in the young mice, and not in the old mice, we naturally thought, well, maybe there's some epigenetic modifications that are occurring as the mice mature. And Matt Wilkin at that time, it was prior to ATAC-seq being able to assess open chromatin, but he worked with the lab of John Stamatoianopoulos, and we did DNA-seq, and we were able to then look to see in the chromatin where we saw open chromatin and whether that closed chromatin was in fact potentially limiting our ability to stimulate the Müller glia to regenerate in the old mice. And sure enough, Matt did find that there were differences where in the glia, so normally around the rhodopsin locus, you can see lots of accessibility near enhancers and promoters, and same for NR2E3 and Anand's favorite NRL. But in glia, these things are really shut down. In the adult glia, we didn't see any evidence for open chromatin around a lot of these neuron-specific genes. So we had the idea of possibly inhibiting the histone deacetylase. Histone deacetylase normally represses chromatin, whereas histone acetyltransferase will open up the chromatin and put an H3K27 subtle group on. So Nick Jorstad took this project on to inject the mice with histone deacetylase inhibitors. And so we tried out a number of different ones, both in vivo and in vitro, but we finally settled on trichostatin A because it sticks around the longest. And when he now took adult mice that were 6 weeks, and we've gone even up to half a year old mice, and we inject the NMDA again, but now follow up with TSA. Now, even in the adult mice, the glia go through this change where now we can start to see neuron-like cells, amacrine-like cells, and bipolar cells arising from the Müller glia by the lineage trace. Now, zooming in, you can really see these. And so we track the morphology of these cells. And they survive for many weeks. So we've looked up to 3 months after the NMDA treatment. And they primarily look like bipolar and amacrine cells. I would say probably 80 to 90% look like bipolar cells. With Rachel Wong's group, we did high resolution imaging of these to show that they were had evidence of synapse formation with other cells. We also, with her group, did serial block face EM. And again, this is all published, so I'm going to go through it rather quickly. But basically, we could see that some of these cells were putting processes and interacting with cone pedicles. But we worked with Frederici to do the electrophysiology, and that was really the most satisfying. He made retinal slices from these mice and was able to patch onto cells that were GFP positive, derived from the Müller glia. And this cell shown in the middle, this GFP cell here, was a Müller glial derived bipolar cell. And you can see that it's got a light response here that's very similar to a normal bipolar light response. Again, the morphologies were somewhat unusual, but Fred would say that they had input resistance that was much more neuronal. They had rest potentials that were like neurons, and that they would respond to light. And obviously, we saw both on and off examples. And so that was really gratifying to see that not only can you make neurons from Müller glia, but they will actually integrate into the circuit and in an adult retina, which really I think was pretty surprising, but gratifying. But single cell RNA-seq was just becoming, a thing at that time. And so we also did single cell RNA-seq on these reprogrammed Müller glial cells. And what we found was quite interesting, that they're both, we could find the cells that stayed wild type, even though they expressed the transgene and would associate with wild type Müller glia. We could find a group of cells that were more progenitor-like and expressed like S6 delta 1 and some of these progenitor genes that are downstream of ASCL1. And then we could find a group of cells in the UMAP plots that had more bipolar and amacrine-like states. And so again, the gene expression was more consistent with their differentiating to neurons. And this was nearly identical to what we actually saw from the GFP labeled cell populations within the retina. So to conclude at this part of the talk, I will say that these epigenetic factors normally seem to limit regeneration in the adult mouse, but HDAC inhibition can remodel this epigenome of the glia and allows this regeneration, and the regenerated neurons will wire into the existing circuitry. But if you look at this, the number of cells, look at what we're seeing here. This was a pretty typical experiment. It's pretty typical today if we do exactly this protocol, and we keep running this protocol as our baseline, but this is about the distribution that you see. Some cells stay Müller glia, some cells go into a progenitor state, and some cells differentiate into neurons. And it's actually pretty similar to what the bird retina looks like, that most of the cells really stay in this progenitor state. So that puzzles us a little bit, and we have some ideas of why that is with ASCL1 alone, but we want to improve the efficiency, and we want to improve the efficiency of cells that are in the Müller glial state still, despite expressing the transgene. So we want to both make cells that go into this state, and then cells that go from the progenitor to the bipolar. So we want to improve this significantly. Also, why just bipolar and amacrine cells? Why not other cell types? And again, we've been working on that problem as well. And I'm going to share with you some data that at least we know additional factors can improve both of these issues. Okay. So the first thing I'm going to tell you about is, what about other epigenetic factors? Do they limit regeneration? And I think they do. We started out by asking, well, okay, what if we did an ASCL1 chip-seq on the glia and on the progenitors and compared, whether or not progenitors and glia are really the same at where ASCL1 is binding in the genome. It's naturally expressed in the progenitors, so we're looking at its normal profile, but in our glia, we're overexpressing it. So we compared a chip-seq from both a P0 retina and compared that to a Müller glia. First, we did chip-seq on normal retina because it hadn't been done before, and figured out how it compared with spinal cord and neuroprogenitor cells and other places in the literature. And there's a common core group of ASCL1 targets, some of which I've mentioned, but then there's also a big group of retinal specific targets for ASCL1, and those help direct this proneural gene into generating retinal specific cell types, we think. So we compared with Müller glia, the ASCL1 chip-seq, and you can see there's some peaks that are present in the Müller glia that aren't present in the P0 retina. And there's some peaks that are present in the P0 retina that aren't present in Müller glia. So we find both things happening, that the pink here is the progenitor ASCL1 binding sites, and the green is the Müller glial binding sites. And ideally, these circles would be completely overlapped. But obviously, you can see they're not. There's lots of peaks that ASCL1 is binding in Müller glia that we would call ectopic, or not inappropriate sites. And there's clearly P0 retina peaks, progenitor peaks that are not being bound by the Müller glia. But is when we overexpress ASCL1. So we wanted, the goal of trying to merge these two circles in the Venn diagram into one is really what was driving this next few slides I'm going to show you. we looked at what motif was highly represented here, that was also that was in these peaks. That so ASCL1 was bound there, we know, but also other transcription factors are bound in some of those same peaks. So what are the other transcription factors? And we found that in these regions that are unique to the Müller glial cells, STAT135 binding site was actually overrepresented in 12% of the regions. So that got us thinking, well, maybe STAT is inappropriately somehow opening up sites in the Müller glia that then ASCL1 is binding to, and that may be driving expression of genes that are not normally driven in progenitor cells. And in fact, we actually found that there were STAT sites near peaks like ID1 and ID3, which are inhibitors of neurogenesis. So that got us thinking that maybe the STAT signaling is somehow inhibiting the neurogenesis that occurs from the Müller glia. So we did an experiment where we did the same thing, did NMDA, TSA, but now we also injected a STAT inhibitor, and Nick Jorstad and now was joined by Levi Todd, and they both did this experiment. And now you can see we're getting really effective neurogenesis. Most in this field, there's hardly any Müller glia left. These guys are all forming neurons. And this we published last year in Cell Reports if you want additional details on this. So that was gratifying that in fact we could go from these epigenomic data and then go back, make a prediction, and then test it. And indeed, that turned out to be an important contributor. The efficiency was now getting high enough, really approaching 60%, as opposed to the 20 to 30% that we were seeing before, that now we also undertook another experiment. We decided in the past, we hadn't really been able to see a lot of EDU labeling of these new neurons. And while the lineage tracing is good, we did know one of the most effective ways to show that these are new neurons that are generated in the adult retina is by showing that they'll incorporate EDU during their mitotic cycle. And that's certainly what we did with the bird and what the people in the fish do routinely. So now that the efficiency is high, we ran through an EDU experiment where we injected between days 9 and 12, twice daily IPs, injections of EDU along with the TSA and the STAT inhibitor. And now you can see that there's lots of cells labeled with the EDU, including many of the new neurons that are generated are EDU positive. And it's really, to our eye, it's starting to look a lot more like what you see in the fish regeneration, where now you see both the Müller glia have dedifferentiated to the point where they turn on progenitor genes, they re-enter the cell cycle, they make multiple rounds, or at least several, some rounds of division, we don't know how many, and then they differentiate into bipolar cells and mostly bipolar cells and some amacrine cells. And so this was really nice because now we can say we had another check that in fact the neurogenesis is real and coming from the Müller glia. And here you can see that the percentage of cells in the STAT inhibitor really goes up. The number of neurons we can see right here is getting pretty high. They're still Müller glia, they're still progenitor cells, but when we compare what we call ANT, which is ASCL1 alone, versus ANT STAT inhibitor or ANT-C, you can see that the number of neurons really increases with the decline in the Müller glia. So again, it's making it more effective to get through the progenitor state and to change from Müller glia to a neuron in this system. And this just summarizes and shows Levi in hiking in the Cascades. Okay, Levi also did another experiment where he looked at the role microglia might be playing. And I really don't want to spend a lot of time on this today, but I'm just going to take you through it rather quickly. Normally during injury, microglia really proliferate after the injury. And in fact, we think this may be what's driving the STAT signaling. There's certainly evidence that the microglia release cytokines that then stimulate the Müller glia STAT signaling. So these projects are somewhat related. And so what Levi did was to take away all the microglia. And you can do this by using a drug called Plexicon. Oops, sorry about that. So you can do this by using a drug called Plexicon. And what Plexicon does is it will ablate the microglia by blocking the CSF1 receptor. And so as you see under a normal diet, you've got plenty of microglia shown here in green, stained by IBA1. But if the mice are fed this Plexicon 5622 diet, now it completely wipes out the microglia. This can have some effect on the retina, but the structure is fine. And the effects, Danny Staben's group has really explored this, are mostly in functional effects on B wave amplitude, but no structural effects. There's no injury to the retina that occurs after this, as far as we can tell. But when Levi did this, again, he went from sort of the ANSI phenotype, where you still see some Müller glia and some neurons that are generated after, with the ASCL1. So we still turn on the ASCL1 and we hit with NMDA and then TSA, and you can see there's a mix of neurons and glia. But again, when you get rid of the microglia, now you get a much more efficient conversion of these progenitor cells, glial-derived progenitor cells, into neurons. So these two treatments, STAT inhibitor and microglial ablation, really do improve this efficiency by which the ASCL1 expression in Müller glia will allow these cells to regenerate neurons. And still, in both of these cases, we've damaged with NMDA. We've also tried light damage, and it's interesting because light damage will allow the initiation of this same response. But in the case of light damage, you also see primarily bipolar cells regenerated. So even though bipolar cells weren't killed in the light damage, these are the cells that get produced rather than any other cell type. So there seems to be a bias with ASCL1 overexpression to regenerating bipolar neurons from the glia, but the environment does play a role, at least in modulating the efficiency of that process. So it doesn't necessarily change the phenotypes by modulating the fates of the cells by modulating the environment, but it does affect how efficiently these things undergo this neurogenesis. But so now that brings me to this last bit I want to talk about, which is, what about other transcription factors? We've really focused on ASCL1. What if we branch out and start looking at other transcription factors? And one of the things we realized is that if you look at RNA-seq data, single-cell RNA-seq data, and these were actually, we remapped them from Seth Blackshaw's data that was published last year, but this is mouse at E14 developing retina. And you can see here are the horizontal cells and amacrine cells. Here are the progenitor cells in this group. And then this cluster is the developing ganglion cells, and a transition population where cells that are progenitors postmitotically, they undergo this transition population to these other lineages, and then they'll differentiate into ganglion cells or one of these other phases. And but what's interesting, when we looked at ASCL1, ASCL1 we've known for years is really expressed in the progenitor population, which is why we started with it in order to reprogram cells to progenitors. But in some ways, overexpressing ASCL1, the cells kind of stay as progenitors. So we thought, well, normally cells will express a different type of transcription factor. I mean, I should say a similar type of transcription factor, but of a different subfamily. It's called 807, the atonal class. And atonal class factors are expressed immediately as the cells are leaving the cell cycle. And you can see here, these are the cycling progenitor cells. These are the S phase, and these are the M phase. And just as they exit the cycle, they start turning on 807, and they'll express it through this transition phase and then turn it down as they differentiate into any of these other lineages. So we asked the question, well, what if we expressed 807 instead of ASCL1? And we did that, but in fact, we've done it both in vivo and in vitro. And in vitro, I'm not going to talk about the data right now, but it's similar to what we see in vivo when we overexpress 807 in vitro. But in vivo, we had to use 801 because we didn't have an 807 mouse, but the 801 mouse works very similarly. And so the way we did the experiment was just like what we did with the ASCL1, where we use a Cree from either the GLAST or RLBP, and we drive TTA, and that'll bind to the teto on ASCL1. And we cross that onto a teto-ATO. And now we run the same experiment. We give tamoxifen, NMDA, TSA, and we see what happens. And whereas in the ASCL1 alone, many of the cells remain as Müller-Glia, large progenitor, and then to bipolar and amacrine. With ASCL1 and ATO, these were the Müller-Glia that are left. So it's practically no Müller-Glia are left in the population of cells that express this transgene. Now, we're only expressing the transgene in 10% of the glia at best, probably more like 5%. But of the glia that express it, because we fax purify, there are just almost no glia left. There are still progenitor cells. A lot of the glia do transition into the progenitor population, but mostly they make neurons now. So we've really efficiently driven the process through the progenitor state and into these neuronal cell types. We see amacrines, bipolars, we see cone-like cells, and now the biggest population are ganglion-like cells. And you can really see this from here. Here's our standard ASCL1 overexpression. We see, lots of cells there are still some glial-like cells and processes that go all the way, but lots of neurons. But now with the ATO and ASCL1 expression, they're pretty much all neurons now. Like I said, it's 90% conversion on the single cell, and we basically see that it's very similar on the histology. They express markers of these different types of neurons, primarily HUCD, a marker of amacrine cells and ganglion cells. We can see that here. There's lots of examples of HUCD positive cells. But some will also express OTX, though really OTX is expressed by most of the neurons in the ASCL1 alone, and now HUCD is expressed by most of the neurons in the ASCL1 plus ATO. So we've really shifted the differentiation of these cells. So we asked what these bipolar cells were really clear in their phenotype. They map exactly where bipolar cells should in normal RNA-seq data. Same for the amacrine cells, same for the cones, lots of markers in the cones. But this population, we weren't quite sure what they were. They looked like ganglion cells from the point of view of the HUCD expression, but amacrine cells also express HUCD. So we took these cells and we mapped them onto E14, developing mouse E14 retina from that same data set I showed you earlier. And we can see now that the bulk of these cells really cluster with this population. And what that population is it's this populations are the, this population is the 801 or 807 expressing transition population, and these are all immature ganglion cells right here. So these things are really nicely mapping primarily with immature ganglion cells. And in fact, then when we look at other markers for ganglion cells, in addition, like GAP43, neurofilaments, maybe not as highly expressed, but other genes, SOX11, SOX4, other genes that are known to be expressed in ganglion cells, we see them enriched in this population. And these cells now start to put out nice long axons. And I should also say that we've done the EDU labeling studies, and many of these cells in our paradigm are also EDU labeled. So we know they're newly generated, they're taking on an immature ganglion cell phenotype, and they're putting out nice long axons. They definitely don't go to the brain. I'll just tell you that already. They don't necessarily even follow the normal axons in the optic nerve, but and many of them are displaced into the internuclear layer in the sort of amacrine layer, but yet they still make these axons that go quite a distance. So basically, I just want to wrap up and sort of review a little bit about what I've said. So HDAC inhibitors along with ASCL1 really were able to give us the first evidence of functional regeneration in adult mice in vivo. And the fact that they wired into the circuit was, we've seen that from the very first, and we've continued to do that in the STAT pathway inhibition as well. Fred recorded from us with this last round with the ATO experiments. He's recorded again on some of these retinas, and he sees little spikelets. So this is the first time we've seen action potential in these cells, but that would again be consistent with the immature ganglion cell state. The regeneration occurs, we think, both by direct transdifferentiation and by new neurogenesis. The reason we can't prove transdifferentiation, that's a hard thing to prove because it's the lack of EDU, and we're only labeling with EDU over a particular window. But we know that at least a third of these cells are newly generated presumably by these progenitor-like cells that the Microglia turn into, or by directly stimulating mutant proliferation with this approach. It seems either one of those is still possible. And that remarkably, adding a second proneural transcription factor really directs the cells from being in this sort of proneural neurogenic state of a progenitor through that state and into differentiating neurons. And so we are still, taking some of these studies out longer to see how long these cells last. But in general, from the ASCL1 alone, they would last for many months. I should also say we tried ADO alone, and there it's toxic. So the combination of the two turned out to be important. And so getting this robust phenotype required the two transcription factors together. The other interesting thing is it no longer needs injury. So putting ADO1 and ASCL1 together will generate these neurons at the same rate and roughly of the same types without even injuring the retina. So I think that actually is nice, and we don't need the TSA anymore either. So that having a really powerful transcription factor combination like that seems to be able to move the Microglia through the progenitor state and into a neuronal state, even without adding some of these other factors. So that's also very interesting, and I'd really like to explore why that is. But just coming back really briefly to diseases. So we can, in mice, put some bipolars back into the retina. And I think now by adding another transcription factor, shift some of these, or shift the majority of them to ganglion cells. We also see some cones. So I think we can push the progeny of the Microglia around a bit with transcription factors and transcription factor combinations. But, I would say years ago when we started this whole process, you would think, wow, if you could only regenerate the retina in the mouse like you do in the frog, or like you do in the fish, wouldn't that be wonderful? And I think it is wonderful, but it's also not obvious exactly what disease you would want to target, because obviously there's a lot of genetic diseases that you still have to correct the genetic defect. Glaucoma, you still have to get those axons all the way back to the brain, and that's a big lift at this point. I know people are working on it. But it's still the human brain, the optic nerve length is enormous. So I really would love to hear from people as to what would be the favorite disease target, because from a clinical point of view, I just really don't know. I mean, maybe trauma, maybe detachment. I know the retina, gets bashed around, and people are always writing to me by email saying, my kid has this or that, and the retina is really thin, can you regenerate it? But I think, we're at the point now where this approach of, I think George Church recently called it self-fate engineering. I think we're at the point now where if this isn't regeneration, it's self-fate engineering, and we can do it. And so I think we're just going to get better at it. But the question is, what do we do with that? And so, yeah, I'd love to hear any comments people have, because it's been fun, but it would also be great if it could help somebody someday. So with that, I just want to end by thanking people in my lab. Levi did the lion's share of that work, but was really supported by Marcus Hooper. And also Connor Finkbeiner is our guru for all our data analysis. And Alex Hagen and Wes Jenkins also have played a really key role in some of the work that I presented today. And Nick, none of this would have been possible without Nick Jorstad really taking the lead on this years ago, and Yumi Ueki, who also really got the ball rolling, and screened through lots of mice till she found one that would work. And of course, my collaborators, Fred and Rachel, who was just elected member of the National Academy, so congratulations to Rachel, and and my funding, the I Institute and the FFB, and more recently, Genentech and Open Philanthropy. And thank you for your attention, and I will stop sharing. Okay. Stop sharing.
Thank you so much. As expected, wonderful presentation. So we have questions, and if you have one, just please send a quick chat. Tom will have to leave, unfortunately, not that long from now, but we still will extract from him all juice he has. So
and anybody who has a question, if we can't answer today, just send me an email. I'm happy to.
All right, Dorota.
Hi, Tom. I'm sure you expected the question from me. So we met first time in 2004. I don't know if you remember, but it was about MASH1, ATO7, and Neurogeneration 2, and all this discussion. So one of the things which I have plenty of questions. Maybe I will just start discussion with you by email, but I have one which just, I think the data I've seen first time, the ATO7 and ATO1. And so actually, you know that ASCL1 is never co-expressed with ATO7, but it is co-expressed with ATO1. I think it was pretty smart to use ATO1 in this particular situation. It is expressed in the ear, right? In the ear development.
Right.
So this is pretty interesting that you got those ganglion cell-like cells. So it's probably this is a way to go for axons, long axons, right?
Right.
But I wonder, how would you design with ATO7? Or maybe what would happen if you know, or you think, if you changed instead of ASCL1 to Neurogeneration 2?
Yeah, that's a really interesting question. We tried Neurogeneration 2 years ago, and it was like a weaker version of ADOS or of ASCL1. But I would say it was before we were really doing, the single cell. We didn't have that single cell resolution. So we just, all this stuff was started with just taking the whole culture and running PCR for a few genes, looking for rod gene expression or cone gene expression or something. And so the resolution was terrible. You'd have to have gotten a massive conversion in order to see anything from PCR of 500,000 cells. So I would say it would be worth revisiting. We do know that the 807 in the cultures now, we've done the single cell with that. And it is slightly different than the 801. So you're right, 801 is giving a slightly different phenotype. This paper by George Church just recently came out where they expressed every transcription factor in the in ES cells, and 801 really rose to the top as the best neurogenic factor, as the best proneural factor for ES cells to convert to neurons. So there are clearly differences among these transcription factors in maybe the cofactors they recruit, the exact motif that they bind to, things like that. So I think it will be possible to find the perfect combination to really dial this in. But I think that's the challenge now is, now that you can do it, the question is finding the exact mix for the cell you want. And then, so you don't have to do it the way the fish does of making everything and then, selecting the ones, by death or by, however they prune it down. I mean, the fish regenerates all of the neurons from any injury, regardless, there is some bias, as Jeff Baum pointed out in one of my talks, but clearly Dan Goldman says that not much. So the fish, just any injury will generate the whole thing. But I think in the future we might, as we learn better how to direct these cells down to particular fates, we can create that bias to selectively repair. That would be the goal, I think, now.
Exactly. And then we can look, for example, for glaucoma
Right.
approaches when we lose the ganglion cells and directly make ganglion cells and try to direct them to the optic nerve.
Yeah.
Thank you so much.
Okay.
I have
And
plenty.
I
I will catch up.
Shiming had a good question.
Yes, Shiming has a question, please.
Yes, so there's two questions. Shiming, do I generate rods? That seems to be the hardest thing to regenerate. We put every transcription, in fact, I think we got some of these from you, and we put every transcription factor in the book that is known to be expressed in rods, including, which ROR beta isoform. And man, we really, we repressed VSX2 with the nominal negative, all this kind of stuff, and really never get rods. So rods tend to be really special. we didn't get a lot of cones either, and the chick doesn't get cones, but now we're starting to. So so that's interesting. Why cones and not rods? We did try blimp, and of course, and so we did. Yeah, so we've really hit the rod thing hard, but really come up short. But I think cones are going to be are doable now. So subtypes of bipolars now, the diversity is it's great. We see at least 5 or 6 different subtypes of bipolars. So I think, yeah, I would say there's some resistant phenotypes, but mostly it's a question of relative proportions. And again, if you've got a cone disease, you don't want to generate mostly ganglion cells, so you get a few cones. So I think there's a lot of work to be done to really understand this. And also, you know, this is early days. Everything we put in, we're surprised by. So obviously, in terms of understanding the actual mechanisms as to what's binding where and how and who's turning on, you know, who knows? I mean, again, there's lots of work that needs to be done to figure out how to make this closer to developmental biology now.
Tom, Dorota had a follow-up question. Dorota.
About blimp. Yeah, no, blimp, we also tried PRDM1 because Joe Brzezinski had shown in our lab years ago, developmentally, that if you knock out PRDM1, you convert the bipolars to, or you convert the photoreceptors to bipolar cells. And so,
and it's very interesting factor because it's actually the chromatin reorganizer. So it's not particular. Yeah.
No, and it's interesting because it's a transient factor. We think it's, even when Joe was in the lab, we thought microRNAs are knocking down. In our dicer knockout, we saw a blimp going way up in some of those cells. So we do, I mean, it would have been perfect, but it didn't work. You know, and these are negative results. I mean, so you just go with what works when you're in the reprogramming business. But I think I think we could probably do it better, like all these things.
Have you thought about microglia?
Okay.
Microglia being the source of the regeneration. Have you thought about microglia?
Yeah, so Levi's microglia knockout really did show that they were repressing this neurogenesis. And so that paper was just in December in Cell Reports. You should have a look at it. It's really interesting. He's following up on that. I think he wrote a K99 to really explore microglia, and other immune cells are coming into the retina. I think Danny Zabin's really shown that. So what are the roles of some of these other immune cells? It was surprising just because in the fish, if you knock out the microglia, you don't get regeneration. So Levi thought that's what we were going to see. And in fact, we saw exactly the opposite. We got way better neurogenesis from the glia when you knock out the microglia. And so, you know, I guess the fish, there are really key differences in the way the fish is working and the mouse. And I think, you know, Seth Blackshaw's group has done a nice comparison. And so we can start to take that apart. Dan Goldman's really had a great talk at Arvo on notch signaling. And it's really different it's like literally the opposite of what happens when you inhibit notch in mice versus when you inhibit notch in fish. So there are some really, you know, key regulatory nodes that are, you know, have been altered through evolution that are we can play with, I think. So yeah, anyway, no, I think it's a fun.
All right, let's move to Zach.
So that was actually like a perfect segue to my question, kind of relating to what Dorota brought up with the microglia.
Mm-hmm.
So is it, or could rod regeneration be more dependent on exogenous cues from other cell types in the microenvironment, as opposed to internal, you know, transcriptional factor activation and like very precise timing of cell types entering that microenvironment and secreting factors or ablating specific cell, like progenitor types?
Yeah, no, I think that's a really interesting point. Years ago, we looked at rods developmentally for factors that might promote rod development because it was one of those cell types that we could explore, and we did find that rods, you know, there is some combination of intrinsic and extrinsic factors that regulates the timing of rod development. And so absolutely, if these factors are going to be important, it would be rods would be a likely case where that would be the case. Retinoic acid, it will promote rod differentiation, and there's several other factors that will do that. We've tried those in our in vitro cultures to add those factors. Those things work in developing retina. They don't work in these in the glia, in the reprogramming in vitro. But I think we, again, those are hard experiments to do perfectly. A lot of these factors, if you just, like you say, if you put them on all the time, that's not going to work. You have to do sort of, and that's been known for lots of these signaling pathways, is that the devil is in that detail of timing, and that's a really hard thing to control in vivo. And yet, we have made progress by injecting these factors like STAT inhibitors and TSA, and they do have effects. So I know the environment is important. And so it might just be a question of getting that right environment. So we've talked about that, injecting retinoic acid, taurine, some of the things that are known to promote rods during development.
Yeah.
And
Are ECM factors also at play in terms of, fine tuning adhesion of cell types as they're, becoming more of like a basal progenitor cell type?
Yeah, I think that might be what's different in the fish, is that the fish goes all the way back to this like really early progenitor state.
Oh, okay.
And Dan Goldman has actually shown, so one of the things that we found regulates the early versus the late progenitor state were microRNAs. And so in the dicer knockout, the cells just generate the early fates. They never progress to the late fates if you do the knockout early enough. And one of the things that is important in that is this LET7 Lin28 axis. Now, I know Tim Cherry's now also finding the MIR9, and we had identified MIR9 and LET7 as being some of the critical factors, and MIR125 as being some of the critical factors regulating that early to late transition, because those were affected by the dicer knockout, and we could somewhat rescue it through overexpression of those factors, and so of those microRNAs. So we think that that's regulated possibly by the LET7 Lin28 axis. What's interesting is Dan Goldman found exactly the same thing in the fish, that in fish, when you damage the retina and ASCO1 gets turned on, ASCO1 binds to a region of the Lin28 promoter and drives Lin28. And that should antagonize the LET7. And we know LET7 just keeps increasing throughout development of the retina. And that seems to be this thing that maybe is pushing those cells into this late state. So reversing, so in fish, turning on Lin28, reprogrammed, helps reprogram the fish Müller glia to this really early state. Now, it's necessary, but I don't think it's sufficient, because Dan also did an overexpression with Lin28. We've done this too. And that's not sufficient to make this whole thing work. But on the other hand, not all forms of LET7 are antagonized by Lin28. And so there are clearly other RNA binding proteins that bind LET7. There's a lot of LET7 members. It's hard to knock LET7 down. There's a lot of it. And so that actually led us to doing a dicer knockout in the glia, and then look to see what would happen, whether that would help them regenerate. And we found, Stephanie Wohl did this. So she did the dicer knockout. We knocked it out of the Müller glia. And what happens is the retina gets really disorganized. The glia get really odd. It's almost like a retinal degeneration type effect that you see, like with Robert Mark's really advanced retinal degeneration. The glia just gets so weird, but they don't reprogram any better without dicer. So obviously that was a pretty crude experiment, not, sort of like the conversation I had with Anand the other day about, well, if we get rid of all methylation, of course, that's not going to help. You need to get rid of it selectively. And so getting rid of selective microRNAs versus getting rid of all microRNAs, absolutely. I think those are becoming way more nuanced in this control, in these control molecules is really the way to do this right. But I think what we're doing is just like taking shots at the, throwing the darts at the board, and a couple of them hit.
And hoping to land on.
Yeah. No, it's very crude.
That's awesome. Thank you so much.
Sure.
Tom, I will have a few, maybe two, maybe more, maybe less. We'll see how many questions I will generate. But the question I have, you have been pioneering this whole thing of reprogramming since a long, long time ago. And you have done so beautiful cell biology, all of those processes. And you pose the question which disease you will apply. Do you see that this type of reprogramming has a chance, practically speaking, not in a grant that you're going to cure all of blindness, but do you see a path forward for regulatory and whether we know enough, or this RNA-seq just added yet more element of quantitative but extremely complex network of protein interaction, and we're far away yet from understanding how to do it in a medically or pharmacologically.
Yeah, no, that's a fair question. I actually think right now, if we wanted bipolar cells of all types, or not all, but of many types, and we wanted to convert like 10%, or just like, hang on, and we wanted to convert just 10% of the glia to bipolar cells, for example, we could do that. And so if there were a disease, and I don't know that we can do it in monkey, but we're working on human cells right now, and it does seem to be working on human cells. And so we're definitely going to try it out in a monkey and see. So if there's an application for restoring a population of bipolar cells, then yes, that we can do. And we have a particular transcription factor combination. Looks like it'll do it without maybe the TSA and the damage. And so you could basically say, well, all right, if you've got a disease where you've lost all your bipolar cells, for example, and if you could just replace 10% of them, you could reconnect outer and inner retina, for example, then would that benefit a patient? You could probably do that with an AAV virus with 2 factors directed to Müller glia with an RLBP promoter, and and probably get a subset of bipolar cells back. Now, if we did that for other cell types, if we could get that selectivity to just take a small percentage of the Müller glia, say in the fovea, to restore 10% of your cones in someone who doesn't have any, maybe that would work to give those people some 10, 20 years of vision before those cones got sick and died too. So I think, we can think about it like that. We can also think about, well, if they've already lost their cones, maybe get your cones back and then fix the genetic defect as well. So yeah, no, I think that's the kind of thing we need to think of now, is like, how do we do those?
Yeah, what will be the product, if you will, pharmacological product or genetic product?
I think it would be an AAV with 2 transcription factors, and it would be driven by a Müller glia promoter and directed, single injection. Single injection, reprogram some cells, see if the vision is improved, come back, another injection. I mean, I actually think someday, it's not now, it's not with what we have, but someday that'll be the way to go.
But multiple injections with AAV will not work, most likely, to your immune system, right?
Yeah, I think yeah.
So I know that you may be under a family arrest very soon. So let me just ask Yoshi Imanishi for final comments. Okay. Yoshi, all yours.
Okay. Yoshi.
Yeah, thank you very much for sharing the wonderful progress. I'm always, following your work, and so it's very exciting that you can now create cones. And yeah, I think I'm also looking forward to your progress on ganglion cells, which seems to be more difficult to integrate with the rest of the brain. So yeah, thank you very much, Tom.
Thank you, Tom.
Thanks, everybody. Thank you so much. And really, feel free to send me an email if you have any follow-up questions, and I'm happy to respond. Bye-bye. Thanks again for the invite, Chris. Bye, everyone.
Thank you, Tom, as always. Bye.